Welcome to MOOC course on Introduction to Proteogenomics. We have heard Dr. Ratna in last two lectures about large scale data sciences. Today in the last lecture he is going to continue sharing more information about large scale data sciences. Dr. Ratna will talk about proteomic data commons PDC and about its various dimensions such as ownership of the data, the quality management and life of the data. He will also talk about FAIR principle for developing a proteomic data commons. FAIR stands for findable, accessible, interoperable and reusable FAIR. It includes assigning data and patient a UID unique identification which remains same across the world and hence reusable by the users. The importance of management and upgradation of the repository with unique UID and versions with reason. So, to understand in depth, let us now welcome Dr. Ratna for his last lecture.
This is the minimally viable product that I talked about, pdc.esacking.com. This is an alpha program, so it is even before beta, like I said. So, feel free to log in. I will show, I will run you through that some screenshots of how the portal looks. But feel free to log in and see. This is okay. Like I said, this is a minimally viable product. All of the CPTAG data portal data sets that you see, the colorectal cancer, ovarian and breast cancer data sets that are available, where you just download the information. All that is actually harmonized to PDC. So, you could actually explore all of those data sets here to a much greater extent. Just to give you an idea of what goes into the management, there is stewardship, who owns the data. So, once you put it there, it is done. Is PDC the owner of the data or you still have it? That is called stewardship. And then data governance, what is the life cycle of the data, who will, what kind of policies that will guide the open access of that particular data. And then all other things about the standards and the processing and quality management, all those things are attached to the large scale programs. All right. So, we have to represent the data in a certain way, right. If I am getting data from so many different programs, so many people are submitting, how do I represent it as in a common model? So, I need to have a conceptual model. So, this is our conceptual model. So, because all of this is cancer related data to begin with, so this is patient centric. I am not talking about any model organisms and so on at this point, but even from model organisms the data can fit here, it is pretty easy. So, you have a program, project, a case is basically a patient or a donor who gives the tissue, and then from that you get a sample. So, there is a lot of clinical data that is attached to a case, right. So, clinical information is attached to the patient, and the next part I am showing this part right now. So, then you have an aliquot, that is what actually goes into your mass spec, right. So, that from there you generate, so you group a bunch of samples, run it as an experiment or a study, right? And then you have all of the run metadata, that is nothing but your experimental design where we are capturing it, and then you generate the raw files and then we run the workflow. So, when I run the workflow, it generates other information. So, every piece of information that the software generates, we index it. So, it is captured in the model. So, when I ask a question about which proteins are expressed in this particular aliquot or a sample, I have an answer for you, right. That is because we have a model. So, we are working hard to actually refine this. We try to fit all of the CPTAG data in this model and it is working quite nice, but there are a lot of unexpectations that happen, right. So, if there is something that comes up that actually needs change in the data model, then we have to do it, but for now it is working. All right. So, then there is this data dictionary. So, every node in this graph is basically we have a description of it. So, you can go there and try to understand what it is, and each of these terms are actually we apply standards to that. So, for example, here diagnosis and demography. So, these are all clinical terms there. So, are we using certain standards, for example, ICD codes to define the disease or the SNOMED CT terms? All right. So, then I will briefly talk about the FAIR principles. So, when you are developing a data commons, the guiding principles for such a kind of effort are called FAIR, findable, accessible, interoperable, and reusable. So, what is findable? So, like I said, every piece of data that comes in, whether a patient or the file. So, we will assign a unique identifier to that. As we call it the UUID, global ID, and then we attach a lot of metadata to it. So, it is unique in the system, so that nothing will change ever. So, even if there is a change, we will version that. So, why it changed and how it changed, so that you can always refer back to that particular patient successfully. And then there is at the file level, most of the times when you start downloading or uploading files, they get corrupted, right. So, what you start uploading and what is received at the end, you do not know if they are the same or not. So, you think you uploaded some file, and but it's not fully uploaded, it gets corrupted. The same thing when you download and you do not realize that there is some error occur somewhere, and you start processing those files, and it will your software will throw error, but it is very difficult for you to understand why the software is throwing error, right, in the first place. So, that is why we record a lot of metadata that is called MD5 or SHA. These are called checksum values. It is a small datum that is attached to a digital object. So, you generate that locally on your computer, you upload the file, and the receiver, you will give him this MD5 value. So, on the receiver on his computer, he will generate the same value, and compare this value, the what you are giving and what he received is the same or not. So, basically it confirms that the data is end to end transferred completely. So, all that information will be captured as a metadata. So, basically you can find any file or any entity in this model by a particular unique ID and with the attached metadata. And then accessibility. So, who owns the data and what is the life cycle of the data? So, when you first generate the data and put it in a system like in a PDC, so you still own the data until you make it public, right. So, as the user has the authority to tell, okay, this is the time I want to make it public, or my program requires me to publish in a particular journal, then I will have to make it public. So, and some of it can be protected, especially in genomics, we know all the germline mutations are protected and you cannot just download. So, there is a data access committee and you have to go through that whole cycle to actually get the information. And then interoperable. So, interoperability is basically about the control vocabularies, how we are defining the terms, like if you are calling trypsin, are you calling trypsin all the time the same way? So, it is confusing sometimes. So, what people do is the control vocabularies or standards, they will assign IDs to those terms, so that you can just use the ID instead of the word. So, the programmatically we will access that the information. When you use the ID, we will get the information, okay, this is called trypsin, and then that is called metadata. And so, with that kind of information, these are, for example, these are 3 different resources, GDC, proteomic data commons and imaging data commons. If we are all using the same kind of terminology to describe the common terms across these resources, any tool I can use without much hassle. And finally, reusable. So, reusability comes with the standards like that I mentioned. So, for PDC, we use a lot of standards from the Proteomic Standards Initiative. So, PSI is a special body within the HUPO that formulates guidelines, how do you represent the data, and also both the formats of the files and also the control vocabularies that needs to be used. It is not as mature as in genomics, but it is slowly getting there. I think in the next several years, so we will probably have more structured terms that people will start using. So, at this time we are, that itself actually has about 100 plus terms that you can use, but at this time we could only map about 10 of them to the metadata that we are collecting. Just a general example of why we need standards. I just wanted to show you this. So, the plug points. Yesterday I came from US, so I do not have a converter. I don't need a converter, I just need an adapter. So I was looking everywhere, I did not find one. But it's okay, when I go back to hotel, like I'm just charging fully, that my computer is now fully charged. But anyways, so all these plugs, they're actually based on some standards. They're not random, right? Each country has different kind of standards and they're using. So in such kind of situation, maybe you can have a converter or you can have an adapter as a solution. So it's possible because you know those people are using standards. And here on the right, I'm showing an example of how a date is being represented. You can write it in any way, right? So probably it will make sense for some of you, but when you give it to the computer, it will get confused. So then we come up with, there you come up with the standards, right? So now we have some converter plugs, and then we have ISO code. You have to represent the date in certain way. So when you are submitting data to, for example, PDC or GDC, we will tell if you are putting a date, it has to be like this, right? That's an example, simple example of standards. All right, so why do we need standards? Because bioinformaticians who are into bioinformatics, they would actually understand this much easier. So it's very, if the same data is sitting in different forms, it's a nightmare for them. So we just have to, I mean, the idea is like you want to compare the protein quantitation data with the gene expression data that you're receiving from GDC. Thing is like you have two files, so why don't we just compare? But if they're in different formats, if you are calling the different gene names, and you have to write parts or some programs, and there is so much effort, so you waste all your time. All right, so just I put something here to give an idea of different resources. This is not very extensive, just a sample. For example, we use CADSR, the Cancer Data Standards Registry and Repository, for representing all of the clinical data information in the PDC and GDC. So every term has a, as I mentioned in the CADSR. And then we know for the next generation sequencing, we all know there are some very well adapted formats, FASTUs, BAMs, and VCF, FAST, and so on. All right, so proteomic standards. So like I mentioned, there are a few already there. So there is something called, probably some of you might have heard about Miami. So minimum information about a microarray experiment. That came like several years ago. That basically tells it's a recommendation. It's not forced on you. It's just a recommendation. When you are submitting some microarray data to some geo or DB gap, so this is the minimum information you should provide. So because it's just a recommendation or a guideline, if you go to geo database, that is Gene Expression Omnibus, it's a mess. There is so much data out there, but if you want to use it, there are actually papers, meta-analysis people just did correcting the data in geo. That itself is a scientific article, right? So that's not worth it. So the same way, I mean, in proteomics also, they started off with, okay, we don't want to force anybody with standards to begin with, but we'll tell, okay, these are the minimal guidelines you have to follow. So that's MIAPI, minimum information about proteomic experiment, right? All right. So then we already have a lot of formats I will show you on the next screen, and also we have some control vocabularies from the Proteomic Standards Initiative from PSI. All right, so these are some standards like the representation, MZML, MZIdentML. So a lot of things came out, but the widely adapted one is the MZML so far, because each piece of software that you're running, each pipeline, it generates a different kind of output. So it's the field is not as mature as in genomics yet. So, but we are slowly going there. So the expectation is that going forward, maybe in the next several years, all these will be adopted. So if they adopt, like whether it is PRIDE or Pepper Data Labs or Proteomic Data Commons, if you have the same format of the file, you can easily compare two things, is much easier. Okay. So with all that in consideration, we did build that minimally viable product that I mentioned earlier. So this, as you can see, there is only one program right now. That's the CPTAG. We started off with that. We have about this much 6 terabytes of data and this many proteins and peptides identified. So a lot of summary information on the home page. We also have application programming interface. Basically, whatever you do on the UI, you should be able to do programmatically also if you're efficient in the programming. And this is the workspace I was talking about. I will show all this. And this is the kind of common data analysis pipeline that we would implement within the system. And some more screens. All right. So before I end, I just want to touch base on the proteogenomic integration, because that's what it is CPTAC is about and the proteomic data commons is about. So at the system level, when two systems are interacting like a PDC and GDC, what do we need, right? So there are some sequence-centric proteogenomic approaches. So that's where, because all of this information is patient-derived, patient-centric, so there is genomic information available for the patient on the GDC side, right? So you want to use that information to run your searches against, right? So how would you get that information? So in the model that we are developing, we are closely working with a framework team from the NCI and also the Genomic Data Commons and the cloud pilots. We are trying to figure out how to bring that information together so that when I say a patient, like when in the PDC, when I look for a particular patient, it will automatically tell, hey, there is some genomic data available for this patient at this resource, you want it, right? So that way you can get that information and put as input for your proteogenomic experiment, database search. And then the other thing, so that's at the very high level, even before you start your pipeline, right? So then if you actually generate the quantitation data already, so it's in the gene matrix, and then on the GDC side, they already have the FPKM, RPKM information, gene expression. They have the bed files, they have the genomic variants. So you bring them and start correlating them, right? So that means here you are actually comparing the results from two harmonization pipelines. You are not comparing the raw data, you are not doing anything there. You are using the information that came out of the pipelines and just comparing them. So the R examples that you are trying, they are trying to basically do that. And finally, can we see all the peptides that we identified in the genome browser? So yeah, you can download the information and you can generate the bed file and you can upload and do all those things, but we are trying to do that for you. For any given data set, it will be automatically available. So these are some visions. I will show some data what's already there later. Yeah, so the idea is PDC will do some basic analysis for you. So when you go there, you already have all these reports generated. But if you want to change something, the personal workspace I talked about earlier, so you can do all those things in your own workspace without affecting the public side, right? So whatever we provide is actually for everyone.
On that data only, not on the data on which I am working in.
Yeah, so the high level goal is we have all this information on the PDC data portal, which doesn't have a login. You go and just go there and analyze, but you have some data that you want to actually see correlate with what is already there. So you want to analyze your data alongside of the data that's already there. You should be able to do it in the workspace. So you load all your data, provide all the metadata so that there will be tools available for you to do that kind of analysis. All right. So this is one use case we actually got. Somebody asked us, okay, I know there is a genomic information available in a proteomic study, and how can I actually seamlessly integrate all this information? This is the use cases I was talking about. So the way we implemented the systems level is like find all the projects in PDC that have genomic data. That's a very easy way to ask, but we add this, find all the programs that have in GDC that have proteomic data. Like you can ask that question whichever way you want, right, in different combinations. The system should be able to do that. So that's where we are trying to lead to. So some more examples of the same thing. All right. So, I will just summarize here. So, we right now the proteomic data commons is in build phase. So, like I said 6 months ago we built the MVP, we started building the MVP and we released that on at HUPO meeting in October. So, we got some feedback, but in terms of the system, like I talked about all of these things, but basically we will have storage, workspaces, tools and containers, models and orchestration. So, you plan early, what can you do with this information right now? So, if you have data, you have some ideas now, I think I convinced you to what to do. If you have no data, that is okay. I guess some people came to me and said like we are interested, but I do not have any data, where do I start? So, there is so much data out there, you can start looking at that. And where the data will live, at least in the PDC side, we say it is in the cloud, and if you do not make it public, you are the owner of the data.
So, I hope today you have learned that the PDC data portal consists of all the omics data on a single platform with UID given to each data set or patient, which will remain same across the world. It enables users across the globe to access and reuse the data. You also learnt about proteomics standard initiatives or PSI and importance of such initiatives. You also got a glimpse that how difficult it is if the same data exists in different formats. Hence, a converter or a standard notion could play a major role in developing repositories which are accessible to all. We also learnt about proteomic standards being used currently, such as MZML, MZIdentML or MZQuantML and others. So, in the next lecture we will shift topics, we will talk about data independent acquisition and Swath Atlas by another guest speaker. Thank you.